Pharmacology of drugs. A therapeutic drug is any substance that is given to treat or prevent illness. It can be natural or artificial. Pharmacology is the study of drugs relating to their actions, mechanisms, uses, and adverse effects. Pharmacokinetic is what the body does to the drug, whereas pharmacodynamic is what the drug does to the body. An adverse effect is a negative effect that wasn't predicted, whereas a side effect is predicted. Drugs follow a lock and key mechanism where the drug is the key and the receptor is the lock. Most drugs, around 99%, bind to proteins on the inside and the outside of the cell. There are 4 classes of proteins, starting with receptors, which can interact with certain drug molecules. As a drug molecule binds to a receptor, reactions are initiated which stimulate or inhibit regular cellular function. Another type of protein is enzymes, which are located in the cell, where one type of reaction can activate or deactivate or alter intracellular enzymes. Therefore, drug-induced alteration can greatly increase or decrease the rate of cellular metabolism. Another type of protein is an ion channel, which sit on the surface of the cell. These can cause a reaction with changes in the permeability of the cell membrane. Where the drug binds to an ion channel receptor, which is a receptor protein, which is a structural component of the cell membrane. The drug's binding may open or close the ion channels to allow or prohibit the movements of ions into the cell. An example is movement of sodium ions into the cell usually excites the cell, whereas the movement of potassium ions out of the nerve cell inhibits nerve cell excitability and function. Another type of protein is known as a carrier protein, which also sits on the surface of the cell, which carries out a process known as carrier-mediated diffusion. They are found on the membranes of our cells. They allow molecules to pass across the membranes of our cells. They allow a route for large molecules such as glucose to diffuse across the plasma membrane down their concentration gradient. Each molecule binds to a carrier protein, where the carrier protein then goes through a conformational change which deposits the molecule on the opposite side of the membrane. Like simple diffusion, both these facilitated diffusion processes do not require ATP, and will continue until there is an equal solute concentration on both sides of the membrane. Pharmacokinetics. There are 4 stages when the drug enters the body. The first stage is absorption, which is defined as the transport of a drug from the site of administration to systemic circulation. The second stage is the metabolism of the drug, where the main site of metabolism is the liver, which is known as first-pass metabolism. After metabolism, the drug may be activated or more commonly becomes less active. The third stage, which also can be known to happen in the second stage, due to the drug being absorbed in areas of the small intestine, is distribution, which is defined as the movement of drugs from the blood vessels into the extravascular spaces and body tissues where they exert their biological effects. The fourth and final stage is the elimination process, which prevents the accumulation of the drugs and their toxic metabolites. The main organ of elimination is the kidneys. However, some drugs can be eliminated through the bile ducts into the gastrointestinal tract or through the lungs. Subscribe to the channel for more.